The overall goal of the following experiment is to examine the mechanosensitivity of the TRPV4 ion channel. This can be achieved by transforming yeast cells with the TRPV4 gene and examining the hypotonically induced calcium entry through transgenic aequorin using a luminometer. This can also be examined with the electrophysiological approach. TRPV4 cRNA is first generated and injected into Xenopus oocytes. After incubation, oocytes are examined by two-electrode voltage clamp upon the hypotonic challenge. To examine the mechanosensitivity at the single-channel level, suction is applied to the membrane patches excised from the oocytes.
I'm Professor Jinfeng. Previously, the activities of TRPV4 ion channels were largely shown by Fura-2 fluorescence. Methods developed here provide powerful and quantitative alternatives to examine the mechanosensitivity of TRPV4. The first method, we adopted the use of luminometry through transgenic aequorin to examine the mechanosensitivity of TRPV4 in budding yeast. In the second method, with the electrophysiological approach, the properties of TRPV4 are examined after expression of TRPV4 cRNA in Xenopus oocytes. The basic method of oocyte electrophysiology, such as the removal of the vitelline membrane and gig-ohm seal formation, have already been demonstrated in the work of Brown and co-worker in this very journal in 2008. Here we'll show additional steps needed.